Assalamu alaikum. My name is Dr. Rasha Abdel Mahdi. My lecture today about intestinal protozoa. In this lecture, I will talking specifically about amoeba and Balantidium as an example of intestinal protozoa. Let's start. First of all, I will give you a brief introduction about parasite and the type of infectious parasite that can cause disease to the human. So parasite is an organism that lives on or in a host organism and gets its food from or at the expense of its host. Generally, infectious parasites that causing disease for human can be classified to two groups. These are endoparasite and ectoparasite. Ectoparasites involve all the insect that lives on the surface of the host, while endoparasites involve all parasites that lives inside the body of the host. Endoparasite divided into two types. These are protozoa and helminth. In this lecture, as I said, I will talk specifically about protozoa, specifically about intestinal protozoa, and I will talking about amoeba and Balantidium as an example of intestinal protozoa. Protozoa are microscopic, one-celled organisms that live free or as parasitic in nature. Protozoa are able to multiply in humans and also can cause disease. Generally, protozoa transmitted from human to human into two ways. Depends on the protozoa types. So, intestinal protozoan that lives in the intestine of a human can transfer from human intestine to another human through fecal oral route. For example, by contaminated food or water or person-to-person contact. While the blood or tissue protozoan can be transmitted from human to human by an arthropod vector. For example, through the bite of a mosquito or sandfly. In this slide, I will talk about protozoa classification. Protozoa can be classified based on mode of movement and based on the main site of infection. So, based on movement, protozoa can be classified to four groups. These are Sarcodina, اللحميات, include the amoeba, Ceropodia, and example of amoeba, entamoeba. We're talking about entamoeba histolytica. Second group is Celiophora, the ciliates, الهدبيات. The example of ciliates is Balantidium. Third group is Mastigophora, the flagellates, الصوتيات. And the Giardia, Leishmania, and Trypanosoma are an example of Mastigophora. Fourth group is Sporozoa, البوغيات, the organisms whose adult stage is not motile. Example of this group: Plasmodium and Cryptosporidium. According to the main site of infection, protozoa can be classified to three groups. First, intestinal protozoon. البروتوزوا التي تصيب الأمعاء. The example: Amoeba, Giardia, and Balantidium. Urogenital tract protozoa الكائنات وحيدة الخلية اللي تصيب الجهاز أو القناة البولية والتناسلية. Example: Trichomonas vaginalis is the important pathogen. Third group, blood and tissue protozoa. الطفيليات وحيدة الخلية اللي تصيب الدم والأنسجة. This group are varied, consisting of flagellate such as Trypanosoma and Leishmania, as well as sporozoans. Example: Plasmodium and Toxoplasma. Now I will move to talk about amoeba as an example of intestinal protozoa. And start with morphology. Amoeba belongs to Sarcodina class. The most important feature that separate amoeba from other group of protozoa is the presence of pseudopods, as you see in this figure, in the trophozoid stage. Pseudopod is a temporary arm-like projection of an eukaryotic cell membrane that are developed in the direction of movement, filled with cytoplasm, used for motility and ingestion. Generally, there are two forms or two stages in the life cycle of amoeba. These are trophozoids. Trophozoids are vegetative delicated radials formed in the activated feeding and motile stage in the life cycle of certain protozoa. Second form or second stage is cysts. Cysts are resting stage in the life cycle of certain protozoa, which is equipped with a protective thick cell wall that allows cysts to enter the outside environment with the passage of feces and remain viable for long period of time. In this slide, I will talk about general amoeba's life cycle. This figure summarizes typical fecal oral life cycle of protozoa. The life cycle of all in the intestinal amoebas are similar. Generally, amoebas transfer to humans through the ingestion of the cyst infective stage in the contaminated food or water. In most instances, Trophozoites are easily destroyed by the gastric juice of stomach and also very susceptible to the environment outside of the host. Due to these reasons, trophozoa are not usually transmitted to humans. After the cyst swallowed, the excission happens in the ileocecal area of the intestine. Excission is the stage in the life cycle of a parasite in which trophozoid escape from a cyst after being swallowed by its host. After excission, replication only occurs in the trophozoid stage and is accomplished by multiplication of the nucleus via asexual binary fission. Finally, when the environment becomes unacceptable for continuing trophozoid multiplication, the encystation occurs in the intestine and cyst will pass in the feces. Encystation is a process in the life cycle of protozoa in which trophozoid becoming enclosed or withdrawn in a cyst. This can be in response to adverse environmental conditions. This slide shows amoeba species. What are amoeba species? Amoeba species are: first, Entamoeba histolytica; second, Entamoeba hartmanni; third, Entamoeba coli; fourth, Entamoeba pollicae; fifth, Endolimax nana. All of these species are non-pathogenic or cause asymptomatic infection, except Entamoeba histolytica. Therefore, in my lecture, I will focus about Entamoeba histolytica. Other species, as Naegleria, Fowleri, and Acanthamoeba species can cause disease, but not in intestine. Now, I will move to Entamoeba histolytica. Let's start with Entamoeba histolytica. Firstly, classification. Entamoeba histolytica belongs to subkingdom Protozoa. Phylum Sargomastigophora. Class Lobaceae. Order Amebia. Family Sarcodina. Genus Entamoeba. Species Entamoeba histolytica. Still continuing talking about Entamoeba histolytica. In this slide, I will show you the important properties of Entamoeba histolytica. Let's start. Entamoeba histolytica has two stages in its life cycle. They are, as you see in the figures, trophozoid and cyst. let's know what is trophozoid and what is cyst. The motile amoeba trophozoid form exhibit rapid unidirectional progressive movement achieved with the help of finger-like hyaline pseudopods. Chromatin bar absent in trophozoid. The cyst. Non-motile forms. It is spherical to round in shape, smaller than trophozoid, contain chromatin bar. Trophozoid is found within the intestinal, extraintestinal lesion and in diarrheal stool. The cyst predominates in non-diarrheal stools. These cysts are not highly resistant and are readily killed by boiling, but not by chlorination of water supplies. They are removed by filtration of water. The cyst has 4 nuclei, an important diagnostic criterion. The mature trophozoid has single nucleus with an even lining of peripheral chromatin and a prominent central nucleolus caryosome. Let's move to another knowledge with Entamoeba histolytica. This slide shows the life cycle of Entamoeba histolytica. The organism is acquired by ingestion of cysts that are transmitted primarily by the fecal oral route in the contaminated food and water. There is no animal reservoir. As you see in the figures. The ingested cysts differentiate into trophozoid in the ileum but tend to colonize the cecum and colon. The trophozoid invade the colonic epithelium and secrete enzymes that cause localized necrosis, and little inflammation occurs at the site. As the lesion reaches the muscularis layers, a typical flask-shaped ulcer forms that can undermine and destroy large area of intestinal epithelium. Progression into the submucosa lead to invasion of the portal circulation by the trophozoid. The most frequent site of systemic disease is the liver, where the abscess containing trophozoid form. When the condition becomes unacceptable to trophozoid, the trophozoid will insist and pass to the outside in the stool. This slide shows the typical procedures that use in the lab to diagnose Entamoeba histolytica. There are 5 ways to diagnose Entamoeba histolytica. First, macroscopically. Macroscopic identification of cyst and trophozoid in the stool. Sample could be fresh stool, weight amount, with or without iodine stain, or concentrated from fresh stool. However, this procedure are not useful for demonstrating trophozoite. Aspiration or biopsy. Sample obtained during colonoscopy or surgery. Second procedure, serologically, such as IFA, immunofluorescence antibody. Immunofluorescence antibody procedure is a traditional laboratory technique that utilizes fluorescent dye to identify the presence of antibody bound to specific antigen. The immunofluorescence antibody is very useful to detect the serologic response of patient who has been exposed to certain infection agents. The second technique or serological technique is IHA, indirect hemagglutination assay. This assay detects antibodies specific for Entamoeba histolytica. The antigen used in indirect hemagglutination assay consists of a crude extract of actinically culture. Third technique is ELISA, enzyme-linked immunosorbent assay, is a plate-based assay technique designed for detecting and quantifying peptides, protein, antibody, and hormones. In an ELISA, antigen must be immobilized to a solid surface and then complexed with an antibody that is linked to an enzyme. Third way for diagnosis Entamoeba histolytica in the lab is isoenzymes. Fourth procedure is monoclonal antibody antigen detection. Monoclonal antibodies antigen are antibodies that are made by identical immune cells that are all clones of a unique parent cell. Fifth way for diagnosis Entamoeba histolytica in the lab is polymerase chain reaction (PCR) test. Polymerase chain reaction test is a method widely used in molecular biology to rapid make millions to billions of copies of a specific DNA sample. It is fundamental to much of genetic testing, including analysis of ancient samples of DNA and identification of infectious agents. In this slide, I will talk about disease and clinical findings that caused by Entamoeba histolytica. Entamoeba histolytica can cause three forms of disease. A. Intestinal amoebiasis. It begins with abdominal discomfort and episodes of diarrhea of varying duration. At first, mushy, then increasing mucoid, including blood, tinged, so-called red currant jelly stools, dysentery. B. Extraintestinal forms, liver abscess. The symptom is remittent fever, sometimes high, upper abdominal pain, liver enlargement, elevation of diaphragm, general weakness, and other symptoms. Notes: large liver abscesses that are not treated in time are often lethal. C. Other forms of extraintestinal amebiasis are much rarer and include involvement of the lungs, brain, and skin. Let's go to the treatment. The treatment with metronidazole, flagyl, or tinidazole. Asymptomatic cyst carriers should be treated with etoguinol or paromomycin. Now, I will transfer to the second species of intestinal protozoa. And I will start with classification. Blantidium coli belongs to phylum Celiophora. Class Caenorhabdiformes. Order Vestibuliferae. Family Balantiridae. Genus Balantidium. Species Balantidium coli. Continuing with Balantidium coli, this slide will provide you a lot of information about important properties of Balantidium coli. Let's start it. Balantidium coli, the largest protozoan affecting humans, is a ciliate organism, often associated with pigs. These parasites can be transmitted through the fecal-oral route by contaminated food and water. The greenish yellow trophozoid may measure up to 120 times 150 micrometer and are capable of attacking the intestinal epithelium, creating ulcer and causing bloody diarrhea, similar to that of amoebic dysentery. It commonly infects primates, rats, and pigs, and has a worldwide distribution. Balantidium coli infection is mostly asymptomatic, but people with other serious illness can experience persistent diarrhea, abdominal pain, and sometimes perforated colon. The disease is transmitted through transverse spherical cyst, 40 to 60 micrometer. Cyst is infectious stage. Transmitted from host to host by the fecal-oral route. Cyst characteristic by having V-shaped nucleus. Diagnosis of Balantidium coli involves detection of cyst or vegetative form in fecal sample. The figure in the bottom shows the trophozoid and cyst. with macro and micronucleus. In this slide, I will explain Plantidium coli life cycle. The figure summarizes the life cycle of Plantidium coli. So let's start. Domestic animals, especially pigs, are the main reservoir for the organism, and the humans are infected after ingesting the cyst in food or water contaminated with an animal or human feces. The trophozoites exist in the small intestine, travel to the colon, and by burrowing into the wall, cause an ulcer, similar to that of Entamoeba histolytica. However, unlike the case with Entamoeba histolytica, extraintestinal lesions do not occur. Most infected individuals are asymptomatic. Diarrhea rarely occurs. After starvation or an acceptable condition, the trophozoid insists and the cyst pass in the stool. This slide shows the disease, treatment, and prevention of Plantidium coli. What is the disease caused by Plantidium coli? Plantidium infection takes three forms: asymptomatic infection, chronic non-bloody diarrhea, often with halitosis, and severe invasive disease with dysentery similar to amoebiasis. The diarrhea may persist for weeks to months prior to the development of dysentery. The ulcer is similar to that of Entamoeba histolytica. However, unlike the case with Entamoeba histolytica, extraintestinal lesions do not occur. Treatment. Drugs recommended for treatment are tetracycline and metronidazole. And prevention could happen by avoiding the contaminated food and water. In conclusion, parasite is an organism that lives in or on another organism called the host. Protozoa is a unicellular parasite, can infect different sites in the human and cause diseases such as intestine, urogenital tract, and blood and tissue. The most common intestinal protozoans are Entamoeba histolytica and Plantidium coli. Generally, intestinal protozoa transmit to humans through fecal or blood, and their life involves two stages. These are trophozoid active stage and cyst inactive stage. Thank you for your listening. Please, if you have any question, don't hesitate to ask me, and I will be happy to answer you. Thank you.